welcome back. I hope you enjoyed your break and I hope you're ready to really concentrate now and focus on a very important process that is happening right now in your body cells, and that is the process of DNA replication. Before the break, we looked at the cell cycle and we found out that the cell does its normal functioning for 90% of its life. But for 10% of its life, it undergoes reproduction, replication, called mitosis, a simple cell division process. Now, if a cell is going to divide in half, we need to make a copy of the DNA for the new cell. We need to make sure that that DNA is copied precisely, and it's very, very important that you understand why this process of DNA replication has to be so precise and correct. Remember that DNA controls cell metabolism. Every single reaction that takes place in your cell and every single structure in your cell and in your body is made according to the instructions that are on the DNA. Your DNA is like one gigantic recipe book, and each gene is like one individual recipe in that recipe book. And if you're going to make a new cell, it's imperative that that new cell contains all the instructions for how to build the body and how to run that particular cell. So we say that DNA not only controls cell metabolism, but it plays a role in inheritance, because hereditary information has to be passed from generation to generation. And we're not only talking about generations of whole individuals, we're talking about generations of cells. The cells that existed in your body when you were 5 years old are not the same cells that exist in your body today. And those cells, although they have changed over time, the DNA has to be constant. Copies of that DNA have to be made every time a cell divides. We also learned in a previous section that mitosis is the process of cell division, and mitosis makes identical new cells. And of course, in order to produce a whole new cell, we need to replicate the DNA. We're going to be looking at this idea of DNA replication, and I'm going to be giving you a little bit of a puzzle that you need to think about. If we've got the original parent DNA and we need to have DNA in the offspring or in the daughter cell, we've got a couple of options. We could have two brand new daughter strands of DNA produced. Hypothesis 2 says we could have one completely brand new daughter strand plus one parent strand. Or hypothesis 3 says we have daughter strands that are half parent and half new. Now, these three hypotheses were put forward by scientists who were trying to work out how DNA replication takes place. Remember that at the time they were looking at DNA, they couldn't look at it under a microscope and say, that is precisely how DNA replication takes place. They had to use indirect evidence to work out how the replication process takes place. Some scientists said brand new DNA is made. Now, I think you can see the problem with that. If brand new DNA is made, in other words, we're looking at hypothesis number 1, if brand new DNA is made, that means that where did the instructions come from to make that DNA? There's a problem there. If we're going to take all of the DNA and make brand new copies, brand new strands, where did the instructions come from? Well, the instructions came from the DNA. And if we're going to break down the DNA and make new strands, that just doesn't make sense. It's not logical. And so scientists proposed hypothesis 2, where they said we've got the original parent strand And from that original parent strand, we have a new daughter strand produced. Very much like you might take a copy of your notes to a photocopier and you push the button and you've still got your original, and now you've got a whole copy of that original. But scientists actually put forward a third hypothesis, and they said each strand of DNA opened up, and each side of that open DNA molecule became a template for a new half copy of the DNA, and that was where hypothesis 3 came along. So which one is the correct hypothesis? Well, the correct hypothesis turned out to be hypothesis 3. Something called semi-conservative replication takes place, where we end up with two daughter strands, but each daughter strand is half parent and half new. And I think you can see that this is the best way to replicate the DNA without making mistakes, because DNA that has mistakes in it is problematic DNA. And so our copying process has to work precisely. The DNA has to split open and expose nucleotides, and brand new nucleotides are slotted in along the parent strand, and that keeps the DNA replication process precise so that no mistakes crop up. Sadly, mistakes do still happen, and after the break we will look at some of these mistakes, which are called mutations.